And I'm reviewing, of course, for my finals and things like that, but I figured I would teach these things, these concepts that I'm struggling with to you, because if I can teach them to you, then I understand them. So today we're going to be going over some biology stuff, namely photosynthesis and respiration. And I'm going to use my handy dandy iPad here to explain it and draw pictures. But basically this is like a study session, sort of. I'm going to explain how it happens, and then if you're in a bio class or anything like that, photosynthesis, respiration, it'll be on your final, it'll be on your exam. Like, you can't do biology without hitting photosynthesis and respiration. So they're very key, important concepts, and I think it's just helpful to go through it, to review. I can watch this if I want to review myself, but this is my handy dandy iPad. Little tabbies in my handy dandy Apple Pencil. It's very nice, very nice. It's my super long nails. But basically, if you don't know, photosynthesis is the process that organisms use to convert light into energy. So instead of consuming other things, they eat sunlight and they turn that into their energy. Energy in this case being glucose. Glucose, the chemical combination is C6H12O6. So 6 carbon, 12 hydrogen, 6 oxygen, and that's the compound for glucose. But glucose is the sugar that plants and organisms make to eat. Basically, it's their energy. So here I'm going to write that down. The chemical formula for photosynthesis, which is important to know, is that you have to think about what goes into photosynthesis. So what do plants eat, right? Plants eat the CO2 that we breathe out. So it's 6 CO2 plus just light energy. And then they also eat water, right? So plus H2O, the chemical formula for water. CO2 plus light plus H2O. That is the chemical combination for the start of photosynthesis. And plants are going to turn that into, so what comes out of photosynthesis? They get glucose, which is C6H12O6. And then they also get and then they also, of course, give off oxygen. That's where we get oxygen from plants. So, long story short, the chemical equation for photosynthesis that'll focus is carbon dioxide plus light plus water yields, that's what the arrow is, glucose and oxygen. So the way this starts is, in a plant you have these things called chloroplasts, and chloroplasts have these little structures in them called thylakoids. All of photosynthesis is going to take place in a chloroplast. That's where they're going to do photosynthesis. But there are two parts of photosynthesis, light dependent and light independent. As you can guess, the light dependent requires light. The light independent does not require light. The first step we're going to talk about is the light dependent part. So this part needs light, and it occurs in the thylakoid of the chloroplast. So in the thylakoid, we have electrons, right? And I'm going to draw this in a really interesting way, but we're going to have this sad electron. Hold on, that's way too big of a pen. This little sad electron. He's sad, you can see, because he's not energized. But the sun is going to come in here, and the sun is going to energize our little buddy. So he's going to absorb light waves at a frequency of 680. So he's going to absorb that light right there, and that's going to make him happy. So he's going to absorb that energy. He's going to get all happy. And now we have a happy electron. He's energized. He's bouncing off the walls. He's super excited. But as he bounces off the walls, he's going to release that energy that he just absorbed. And he's going to become a sad electron again. But that wasn't for nothing. That energy he just released is going to go to this super fun process called hydrolysis. So hydro, the prefix, means water, and lysis is to break apart. So hydrolysis means to break apart water. So we know that we're getting H2O. It's going to the equation, remember, CO2 plus light plus H2O. is ADP. It's just sitting in there, but these H plus ions that we just got from H2O from hydrolysis are going to go through that tube because there's a concentration gradient. I know there's a lot going on here. Basically, this is happening inside the thylakoid, and there's more H plus out here than there is in here. So it's going to naturally go through that concentration gradient through the ATP synthase, and it's going to add that H plus ion onto the ADP to create ATP. So I know that that was a lot, but we're going to do a quick review. So basically what we've done, we took our sad electron here. He's not energized, but he gets energized from 680 and light. He gets super excited, bounces off the walls, releases that energy. That energy he releases is going to hydrolysis, where we're going to break water apart. We're breaking H2O apart. Those two positively charged hydrogen ions are going to hang out, and our oxygen ions are going to leave. That's where we get oxygen from. Since there's more hydrogen out here and none in here, it's going to go through this protein called ATP synthase. So the hydrogen is going to get added to that ADP that's sitting in there to make ATP. And now we've created ATP. So in this step, we've created ADP and ATP. So that entire process is what's called Photosystem 2. These processes are actually out of order because of the way they were discovered. So Photosystem 1 was discovered first, but then they found out, wait, there's actually a process that comes before it called Photosystem 2. So that was all of Photosystem 1. Now we have our sad electron here, right? Well, guess what? He's going to absorb even more light from the sun, but this time he's going to absorb it at a rate of 700. And he's going to get super excited again, super crazy, bouncing off the walls. Our super happy electron. But then he's going to release that energy from bouncing off the walls. And once again, we have a sad electron. Now, where's the energy going to go this time? Basically, we have this thing called NADPH. So that sad electron, the energy from that is going to go to the NADP and turn it into NADPH. So it's just the happy electron energy is going to NADPH, and that is Photosystem 1. So Photosystem 1, the electron gets excited again with 700 light wave, and then it gets all sad. The energy from it getting sad goes to the NADP and creates NADPH. That, my friends, is the light-dependent reaction. It is light-dependent because it is completely dependent on the light from the sun to come and energize those electrons. So now we're going to move on to the light independent reaction. So I just have to erase all these super fun little notes. I'm going to leave the equation at the top because that's kind of important. So this next part is not going to occur in the thylakoid. It's still going to occur in the chloroplast, but outside of the chloroplast in this place called the stroma. So this next part is going to occur in the stroma. It is circular. So its reactants are its products. It's a big circle. Basically, I think this one's a little more complicated than the other one, but I still think it's totally doable. We're gonna take, so what did we make in the last equation? Well, we created ATP, right? We took the ADP and we made it ATP. So we have ATP, and we also made NADPH. So we have ATP and NADPH. Now, this circular reaction has already been going on, okay? So our ATP and NADPH are going to get added to this thing called PGA. So 12 PGA, we have 12 of them. And the ATP and the NADPH is going to get added to that 12 PGA to create 12 PGAL. Now, this is where it gets a little funky. The 12 is going to break up into 2 and 10. So 10 of them are going to continue on with the cycle, and 2 of them are going to go off. So hold on, 10 BGAL, 2 BGAL. And when it breaks off like this, it's going to release back ADP and NADP. So, and NADP. So, I know, a little crazy. I'll explain it a little more in just a minute. So that 2-BGAL is actually crazy. Glucose is actually C6H12O6. So now we've just made glucose. That 10 BGAL, is going to add to this thing called RUBP. And the RUBP is going to have 6 CO2 added to create 12 BGA again. So as you can see, it's a circle. I'll explain this a little better this time. So from the light dependent reaction, we have ATP and NADPH. It's going to join with our 12 PGAL, right? It's going to join with our 12 PGAL right here our 12 PGA to create 12 PGAL. 10 of it's going to go on to continue the cycle, and 2 of it is going to break off to make glucose. Now when it does that, our ATP and our NADP leave because the extra H there, the H here and the H here to make it triphosphate and NADPH were added to the PGA to make it PGAL. So now they're gone. They can just go on to their next place, wherever they're going now. They're going to go back to feed into the light dependent reaction. That ADP is going to become ATP again. That NADP is going to become NADPH again, and it's going to come back full circle. So that 10 PGAL is going to go back and become this. It's going to have CO2 added and continue the circle. So now we've seen our CO2 gets added. We've made glucose. So let's look at our equation at the top. We've used our 6 CO2. You can see it get added right here. Our light was used in the light dependent reaction to excite the electrons, and our H2O was broken up in the light dependent reaction. What have we gotten out of it? Well, we made glucose right here. So we've made glucose, and we made oxygen when we were doing hydrolysis in the last equation in our light dependent reaction. So basically, I know that was a lot. That is photosynthesis. So to recap, it occurs inside the chloroplast. In the chloroplast, we have the thylakoid and the stroma. Inside of the thylakoid, the light dependent reaction is going to occur. Light excites electrons. That energy from the electrons is going to be used to break apart water. Oxygen is going to leave. Those hydrogens are going to go through ATP synthase and become ATP. That electron is going to get excited again, this time at a frequency of 700. The hydrogen is, that energy is going to go to NADP and become NADPH. That's the light dependent reaction. Now we're going to go out into the stroma outside of the thylakoid, but still in the chloroplast. And our NADPH and ATP are going to be added to 12 PGA to become PGAL. Now we've reduced that ATP and NADPH to ADP and NADP so they can go back to light dependent reaction. Our 12 PGAL are going to break off into 10 PGA and 2 PGA. The 2 PGA is going to become glucose, and that 10 PGA is going to go back to continue on the reaction. It's going to add on to RUBP. That RUBP is going to have 6 CO2 carbon added to it, carbon dioxide added to it, and then it's going to become 12 PGA again, and it'll continue that cycle. So it's cyclic. Its products are its reactants. And that, my friends, is photosynthesis. Light plus CO2 plus H2O yields glucose and oxygen. I hope that made sense. I know that was a crazy crazy ride. But I hope that made sense. Now we're on to the part that I struggle with, and that is cellular respiration. So I have my notes up here to look at if I need help. But cellular respiration is something that organisms do create energy once again. So you can either do it aerobically or anaerobically, with oxygen or without oxygen. But every single organism that does cellular respiration starts with glycolysis. Remember earlier how we talked about lysis means to break down? Well, glycolysis is the breakdown of glucose. So basically, I'm sorry, I have to look at my notes. The equation for cellular respiration is glucose plus oxygen. You breathe in oxygen. Glucose plus oxygen yields water, carbon, and ATP. Our real goal here is to create ATP because that's our energy source. So, glucose, C6H12O6, yeah, glucose plus oxygen yields water, carbon, ATP. That's our cellular respiration equation. But no matter if you're gonna respirate aerobically or anaerobically, you have to start with glycolysis. Glycolysis, the breakdown of glucose. So the way this works is we're gonna take our glucose, which is C6H12O6, and we're gonna add 2 NAD and 2 ATP. This is what it looks like. NAD is that thing we were talking about earlier. 2 ATP is our energy source, so that's gonna like spur on the reaction. This is going to yield 2 pyruvic acid, 4 ATP, and 2 NADH. So that NAD had a H added to it, a hydrogen. And now that we've done this glycolysis, we've broken down glucose by adding NAD and ATP to create 2 pyruvate, 4 ATP, and 2 NADH. We can either go aerobic or anaerobic. So if there's oxygen present, we're going to go down the aerobic pathway. If there's no oxygen present, we're going to go down the anaerobic pathway. We're going to start off by assuming that oxygen is present. We're going to go down the aerobic pathway. So the first part of the aerobic pathway is what's called pyruvate oxidation, which is exactly what it sounds like. So pyruvate is going to be oxidized. So we just made 2 pyruvate acid right here, right? So we're going to take that 2 pyruvate and add this super fun thing called acetyl-CoA. It's abbreviated CoA. So we're going to add some acetyl-CoA, and that is going to yield 2 acetyl-CoA. This is actually just CoA, it's not acetyl. So 2 pyruvate plus CoA yields 2 acetyl-CoA. 2 NADH and 2 CO2. That is the basis for pyruvate oxidation. You can see how the stuff from up here, the pyruvate, feeds into down here, and then we're gonna make this. Yeah, that's pyruvate oxidation. The next step is called the Krebs cycle, or the citric acid cycle. So in the Krebs cycle, we're gonna take the 2 acetyl-CoA that we just made, and then we're gonna add OAA, which I wish I could tell you what the entire word is, but I can't. I just know the abbreviation. This is the really long one, so this is going to yield a lot of things. This is going to yield 2 citric acid. That's why it's also called the citric acid cycle, because it yields citric acid. 2 ATP. Once again, ATP is that energy for 4 CO2, so 4 carbon dioxide. 6 NADH and 2 FADH. So NADH and FADH are just electron acceptors. They accept electrons, they give electrons, they do that kind of thing. So this is what the equation looks like for the citric acid cycle or the Krebs cycle. So glycolysis, we take that 2-pyruvate, we plug it back in, we add our CoA, we get this. We take that 2-acetyl-CoA, plug it back in, add our OAA, and get all this for the citric acid cycle. Now, the final part of this equation is this super duper funky fun awesome thing called the electron transport chain. Let me tell you, I think the electron, like, this is where the magic happens, okay? We've been making all this crazy stuff, all sorts of crazy things have been going on, but now we're finally going to get to use them all. So the first thing that goes into it is 10-NADH. Where are we getting this NADH from? Well, that's my dog. If you look at the previous equations on here, you can see here we have 2-NADH. Here we have 2-NADH, and here we have 6-NADH. 2 plus 2 is 4, plus 6 is 10. So there we go. We've got our 10-NADH. Sorry, my pen is the wrong color. 10-NADH. Okay, so that 10-NADH is going to get split up into NAD plus H. So like I said, it's an electron acceptor. That electron at the end is just there to basically get split off later. So then we're going to take 2 FADH. We got those FADH during the Krebs cycle. So that 2 FADH, same thing is going to happen. We're going to split it up into FAD plus H. I think you can hear the rain. Yeah, you can hear the rain. FAD plus H. And then that is gonna, both of those together are gonna do this super awesome thing. And they are going to create 32 ATP. And that has kind of been our goal all along. It's been to make ATP. And then finally, we have we have some oxygen left over and some hydrogen left over. So basically, the reason that we get all this ATP is it creates a concentration gradient. So through diffusion, it goes through H plus buildup in the outer compartment of the mitochondria, because this is all occurring in the mitochondria, the powerhouse of the cell. The H plus travels into the inner compartment through ATP synthase, and the ADP turns into ATP because of all the H's. And then water becomes the final electron acceptor. So the H ions are going to add on to the oxygen ions to create H2O, or water. Super duper fun, I know. So that is if you take the aerobic pathway. We go glycolysis, pyruvate oxidation, Krebs cycle, citric acid cycle, and then the electron transport chain, all in a super crazy awesome attempt to create as much ATP as we've created. And then if you want to go the other way, gosh, I love notes. If you want to go the other way, you take the reactants. Basically, you take what you get from glycolysis to pyruvate acid and 4-ATP to NADH. Basically, this is what happens when your body doesn't have oxygen. It does this. You can either do lactic acid fermentation, or you can do alcoholic fermentation. So if you ever run or worked out really, really hard and then you cough up some stuff, that's lactic acid, or your muscles get super sore. It's because they're secreting lactic acid because of the lack of oxygen. I'm just going to explain this. I'm not going to write it down. But 2 pyruvate acid plus 2 NADH yields lactic acid and 2 NAD. That 2 NAD is just enough to go back and keep completing hydrolysis or glycolysis. It's just enough to barely sustain your system. Without oxygen, since you're a big, large mammal, without oxygen you don't get through the whole thing. So you're just doing enough to keep yourself alive. You're just doing enough to revert that NAD back into NADH and then do it over and over again. But if you're not a huge mammal, and say you're a tiny little plant or a fungi, then you can do alcoholic fermentation, which is where if you've ever left a pineapple out for a couple of days and then you eat it and it tastes a little questionable, that's because it's alcoholically fermented. The acid is going to add on to that 2-NADH. It yields ethyl alcohol, which is alcohol, 2-NAD and CO2. And then those are just going to go back into the equation and keep just going to do glycolysis, alcoholic fermentation, glycolysis, alcoholic fermentation, and just going to do that over and over again. And since they're such small organisms,